which you guys would have studied in grade 9. You would have talked about homologous chromosomes as being part of mitosis. And then we talked about alleles. Alleles being different forms of the same gene. This is where things are starting to get new. There was nothing in grade 9 that, unless your teacher took things further than perhaps they were supposed to or needed to. This concept of allele here is really the brand new piece. So alleles are different forms of the same gene. So what your hair does, whether it's straight or curly or wavy or whatever, are examples of a particular allele. Or eye color. There are many eye colors, and eye color is actually really complicated. It's more than just a singular allele, whether your eyes are brown or hazel or green or blue. Or I had a buddy in elementary school who had like the halvesies eye, where like it was a line straight up and down the middle, and the left half was brown and the right half was blue. And like eye color is complicated. Most of these traits are complicated. So then we talked about chromosomes, and now we're going to talk about something called a karyotype. And today's lesson, again, is short. It's literally this page, and then the lesson is done. So, karyotype. When you have an organism, and within an organism, they're going to carry, they've got all their chromosomes, and all their chromosomes are outlining their alleles, their gene expressions for different traits. You take all of those chromosomes and you slap them together and you get what's called a karyotype. We could do this for you. Every one of you would have a different karyotype because none of you are genetically identical to anyone else in this class. Unless someone is keeping the secret of identical twins in this class secret, which would be mind-blowing. But not here. Not in this class. See, but your genetic materials, even your genetic material, Numrit, is pretty strong. Because you and your sister side by side, there is no doubt who the two of you are. So, the particular set of chromosomes that an organism can be seen in, the particular set of chromosomes that an organism has, can be seen in a karyotype. Punctuation is important. So each one of you guys would have your own version of this. To prepare one of these, to prepare a karyotype, a cell sample is collected and treated to stop cell division. So you just take any one of your cells that divides. Your upper skin layer wouldn't, but you've got layers, I think it's like the third or fourth layer down in your skin. That's where your skin is created and pushed towards the top. We take one of those cells, treat it to stop cell division during a particular phase. What phase, do you remember from last year, has all of the chromosomes condensed in the cell? Is not anaphase. There you go, Malik. So we were going to get there eventually. You have 1 in 4 chance of getting it right every time we guess. So, we want metaphase. Metaphase is the phase where the nucleus has dissolved and the chromosomes have arranged them, or the DNA has condensed into chromosomes, and they've arranged themselves in homologous pairs. And it's metaphase of mitosis. We don't want meiosis because things are duplicating. We don't want that. This particular sample can then undergo, hands up if you played with the microscopes last year. You should have. You were supposed to. You looked at mitosis under a microscope. Oh, sad. Who'd you have? Oh, yeah. She's scared to work in the lab. She was scared to work in the lab. Don't tell her I told you. But she's coming back. So, if you didn't have a chance to play with the microscopes, maybe I can find us a way to get into the lab, look at the microscopes. But we've got slides that have all the different prophase, metaphase, anaphase, telophase on it. You can see the different phases happening in these cells. You want one that's undergoing metaphase of mitosis. This sample is what we call stained. You put it on a slide. You stain it. The staining process produces a banding pattern on the chromosomes that you can clearly see under a microscope, which is kind of cool. And the chromosomes are then sorted and paired. It would be a ton of work to do this. It would be very difficult to do. But this can be done for any individual. And then they get arranged. And you can see that they're getting arranged in homologous pairs. So homologous pairs being pairs that have the same alleles in the same spot, they have the same size, they have the same centromere location. So you get these two that go together. This is your one pair, your two pair, three pair. They're all labeled here for you. And they have different roles. Again, I don't necessarily know what all of these pairs do, but you've got 22 pairs of what are called somatic. No, not somatic is not the word I'm looking for. Those are somatic cells. I want autosomes. Yes, 22 pairs of autosomes. And then you've got your X and Y. I don't know if I showed you guys this yesterday, but I talk about how the X and Y chromosomes are quite different. The Y chromosome is actually a lot smaller. That's a Y chromosome. And it seems to be deteriorating as generations go on. We don't know why that is. It could leave us in the future with no Y chromosome, which would be effectively the end of the human race, which would be weird. And the karyotype can tell you a bunch of different things. In this very simple example, what was the gender, the genetic gender of the person who this karyotype was taken from? What was the genetic gender? The person was a male. Because of the presence of this guy right here. And so this thing's called a karyotype. And in a perfect world, this is where we would have ended class yesterday, but we didn't. So this is the end of the first set of ideas. We talked about DNA structure, we talked about DNA function, we talked about chromosomes and how chromosomes are what they are, and now we've got karyotype, the collection of chromosomes that effectively is the blueprint for making more of you. Every one of you would have your own one of these. So what I'd like you to do, and I want you to take some time consolidating these ideas. Starting on the next page there's a set of questions. This is the structure and function of DNA, and it starts here, and it goes, and it goes, and it goes, all the way to this page here. This page here is our next set of notes, where we're going to talk about how DNA is replicated. Don't do that, but I want you to do everything up to the bottom of this page here, page 12, or question number 12 here. From the beginning of that DNA structure down to this page here, that is the rest of this class. So this page, page 3, page 4, page 5, and page 6. There's 4 pages worth of questions that I would like you to do. And your market say giver. Yes, Gemini. Yes. You're very welcome.